Good morning, good afternoon, and good evening, everyone, and welcome to the Scientist.com and Biointron webinar titled From Single Domain to Single B, Leveraging Biointron's Expertise for Antibody Discovery Success. I'm Andrew Davies from the events team here at Scientist.com, and I'll be your host today. We're very fortunate to be joined by Dr. Gong Liu, Director of Discovery Services at Biointron, and today he will be discussing two discovery approaches, VHH Phage Display for single domain antibody generation and AbDrop single B cell screening for rapid antibody discovery. Welcome, Dr. Liu. Perfect. And please take it away whenever you're ready.
Thank you, Andrew. Hi, everybody. So first, I need to thank everybody for coming to attend this webinar. I also need to thank Andrew and his team and Scientist.com for what they have done to make this seminar happen. So as slides shows in this title, I'm going to share Biointron's antibody discovery approaches and how these approaches have helped our customer in their antibody discovery success. My talk will cover like these 4 parts. We will begin with a quick brief introduction of my employer, Biointron, and then we'll talk some about the major antibody discovery approaches so far, the most often used. Then we will focus most of the time to talk about Biointron's Phage Display VHH antibody discovery solution, and also Biointron's AbDrop single B antibody discovery solutions. My talk will end with a brief summary. So first, let's take a quick look at what Biointron is like. This CRO company was founded in 2012. We offer mostly antibody production and antibody discovery service, and our headquarters is in Shanghai, China. Currently, we have more than 600 employees, and by this year, August, we've just reached more than 2,500 customers in around 30 countries all over the world. And this slide just give you some pictures about our service facilities, which like DNA preparation, cell culture, protein production, purification, and more. All of those services, these devices, they support our high throughput, fast track antibody production and antibody discovery services. So as I said, Biointron is offering antibody production and antibody discovery services. For the production part, we can offer a 2-week map production, which means from sequences to purified antibody, it just takes 2 weeks. And also we offer fast-track bispecific antibody production and some bispecific antibody pairing and VHH antibody production service, and also some scaled-up antibody production and some cell line services. For the antibody discovery part, so we're going to cover in this topic, which is the Phage Display VHH antibody discovery service and the AbDrop single B cell antibody discovery services. We actually also offer some downstream antibody humanization, affinity maturation services. In addition to these 2 major parts, we also offer like ADC services and offer some off-the-shelf products. Okay, now let's enter our major topic, which is antibody discovery. When it's come to antibody discovery from immune libraries, actually, no matter which choice you rely on, all of the choices kind of generally follow the same workflow. We always start from animal immunization, and then when the immunization was finished, you have to collect the B cells, and from those B cells, you get the very important, most critical thing, which is antibody coding sequences. Once you got those sequences, you are free to go to any of the downstream works like antibody production, hit verification, functional screening. So here I highlight the antibody coding sequences part of the step because this step is the most tricky one. So as many of you may know, I'm not saying it's impossible, but it's very hard to culture primary B cells in vitro. You may catch them for like a few days, but sooner later you will lose all of them. So no matter which approach you rely on, you have to figure out how you are going to broaden this time window that's broad enough for you to get those antibody coding sequences. So to overcome this, so far there are some major approaches. The first one is the well-established hybridoma. So once you finish immunizing in mouse, you can harvest its spleen cells. But as I said, so spleens, B cells from spleens, they cannot survive much long. So what people do is we fuse these spleen primary B cells with myeloma cells. By doing so, we immortalize these B cells. So with those so-called hybridoma cells, we can do, we will have a very broad time window to the any hybridoma screening sequences. After we get the sequences, we can do all of the other downstream work. And the second choice is so-called phage display. So after you finish animal immunization, we also get B cells, we got mRNAs, we got their cDNAs, and then we display these antibodies either as SAV or Fab, or in most cases VHH in phage. Then we will do some phage biopanning to get the positive phage clones. So from this positive phage clones, we will do sequences. Then we get the antibody coding sequences. And a third, which is also kind of a brand new platform, is called single B cell screening. So similarly, you will also harvest the B cells from either mouse or patient. Then you will look at each B cells in a single B level, and then you get the positive B cell hits. So in just like one day or just a few days, you are able to, with this single B cell screening technology, you can get the antibody coding sequences. So these are the so far the most popular three, I'll say, major antibody discovery approaches. So before we begin to introduce the Biointron Phage Display VHH approach, let's take a look at the VHH. So what are VHH antibodies? They are also known as single domain antibody because, so why we call it single domain, because as when you compare to the IgG, you can see the heavy chain antibody, they lack light chain, and also their CH1 is missing. So in addition to its FC part, it has only a VHH there. That's why it's called a VHH antibody. And also because it's a super small, it's a small size, so they are also called a nanobody. This kind of VHH antibody was found from animals like camel, llama, alpaca, and shark. Then you may wonder, since we already have IgG, why do we have to turn to VHH? As most of you may know, this VHH, they hold some significant advantages that make them highly valuable in both research and the biological industry, such as I just mentioned, the small size. This will help them as drug in tissue penetration, and also help them to access some hidden, deep, hard to reach binding site. Second is their high stability because they are solid, small size, and also because of their great water solubility. their high production level. This will favor their drug developability and also help to reduce their manufacturing cost. Third is that they are very easily to be assembled as blocks into many drug modalities like bispecifics, multispecifics, and CAR-T. Last is their lower immunogenicity potential. This is very important in the late stage drug development. This will help reduce ADA risk in drug development. So all of those values have the VHH find broader and broader application in antibody research and development. Okay, now we begin to introduce Biointron's Phage Display VHH antibody discovery solution. Here I list some of the key features that Biointron's Phage Display VHH display solution. First is that in China we have two farms. So we have in-house farms where we farm like more than 400 animals there. So that means we have a controlled animal quality and supply. Second is that we in Biointron we conduct the full pipeline from material preparation to alpaca immunization, PBMC sample collection, mRNA preparation, phage library construction, to biopanning, phage screening, antibody production, antibody screening, antibody validation. All of those, the whole pipeline were done in-house in Biointron. That means so there's a guaranteed speed, guaranteed timeline, guaranteed quality consistency, and you are worry-free of any IP concerns. Third is that actually now every year, or at least in the recent years, we finish well more than 100 VHH project every year. So we have extensive project experience. Our project covers many target classes like mostly proteins, as you seen in the case studies, and some of the peptides and protein complexes and multi-transmembrane proteins. This will also be seen in the case studies, and some small compounds, chemicals, and more. And the most important is that we always follow a target specific solution, which means when a new project comes reach us, we will align the antigen design, immunization strategy, and panning, screening approach well with the target, with the campaign goals, with the target antibodies functional binding requirements. Say we may not always use protein to immunize alpacas. We may turn to mRNA or VLP or cells to immunize animals. And for panning, we may use solid, liquid, cells, or we may combine in one strategy solids plus cells to do the panning. For elution, so similarly, we may not always follow the routine path. We may do some competitive elution, or if you prefer to get some pH-sensitive binders, we may do some pH-sensitive elutions, and so on. And we also have established high throughput antibody production, characterization, and optimization systems, such as plate-based binding, ELISA, FACS, and cell-based function screening, and affinity analysis, and epitope binning, antibody humanization, antibody affinity maturation, and some drug-like trait assessment. Okay, in the coming 5 to 8 minutes, I'm going to show some Biointron's finished VHH project. I will choose some challenging targets to see how our solution support our customers' project. So first one, let's start with well-known targets, which is Claudin 80.2. Many of you guys know this very well. It's a very popular one used for gastric cancer treatment. But this target to raise an antibody against this target was challenging because Claudin 8.2 shows high homology with its sister Claudin 8.1, and also because it's a 4-time transmembrane protein, it has a kind of very small extracellular root showing in the top up. It has 2 loops, 52 amino acid and 30 amino acid for each. So it's a kind of very small size. So which means it's low immunogenicity. And also the worst problem is limited antigen choices, because this multiple transmembrane targets, their protein material is very hard to make. So we have to turn to other choices, like as shown in this case, we immunize animals first with DNA, and then we boost with cells. So as shows in the lower right, after 5 rounds immunization, we got sufficiently high titers. Then after we finish the phage library construction, we do cell panning. So after panning, we did get plenty of good binders that show very good binding to human Claudin 80.2, and they show no binding to Claudin 18.1. The second case is GPCR targets. So as I just mentioned, for these multiple transmembrane targets, this time is 7 times. So it's their protein materials hard to make, and also they are of very low immunogenicity because they are cross species homology and kind of small ECLs. Also very limited antigen choices. But in this case, because this case was done kind of it's kind of new cases. So when we had this project, we already have some better choice like mRNA or VLP. So we turn from DNA or cells to this new form of material to immunize animals. So after 6 round shots, we got high enough titers, and also after phage library construction, we do cell panning. And as shown in this slide, we did got a lot of cell binders that bind very well to both engineered cells and some cancer cell lines. The third case, although it's not this time it's not a multiple transmembrane one, but is a TNF superfamily member. So as many of you know, this kind of protein, they form a homotrimer structure, but the challenge does not lie in its homotrimer structure. Actually, for this kind of targets, they are mostly seen in some immune effector cells like NK cells, T cells. So to raise an antibody against this class of targets, people always want to get some multiple binders. I mean, binders of multiple functions, multiple binding profiles for their early stage comparative studies. Because this kind of binder, their antibody, we don't so far have not yet the best idea how to use these binders for our drug design. Strong binder or fast on, fast off, or fast on, slow off. We have not yet get the best idea. So that means we require a big panel of hits for us to compare and choose from. So to immunize animals, we first prime with Fc-tagged homotrimer proteins, and then boost with HIS-tagged. And then we, for panning parts, we do both solid panning and solution panning. And then we did got a lot of VH binders, and most of them show very good binding activity at both protein and cell level. And most of them also cross-react to cyanotypes and mouse targets. Yeah, as I just mentioned, people may want some multiple choices. So as shows here, our analysis does show that there are multiple dynamical profiles. As you can see, there some of them shows slow on, slow off, or slow on, fast off, or fast on, fast off. So this is very good for our customer to compare and seek the best one of their hits for their drug design. And also, as to the functional part, we also get some blockers, non-blockers, agonistics. So this is very, very good for our customer for their flexible drug designs. And Cas4 is kind of unique because it's not a protein anymore. It's a small compound. It's dasatinib. So it's small compound. So what that means is of ultra low immunogenicity. So we have to, after we cut this small compound into two parts, we conjugate into each part into carriers like KRH or BSA or OVA, and with those conjugate, we immunize animals. After 4 rounds immunization, we got high enough titer, and then we do solid and solution penny. And finally, we got around 30 binders. So the last case is kind of funny, because in many cases, people may want to combine more than one target in one alpaca. So compared to smaller animals like mouse, rabbit, so alpaca is of a much bigger size. So people may wonder, what if I combine more than one targets in one alpaca? So also it can also help reduce the cost. So in this case, we can show that we got high titers for each of the 4 targets combined after just 3 rounds of shots. And also we got very good binders for each of these 4 targets. Okay, after finish this slides, I finished my first part, which is Biointron Phage Display, a single domain antibody solution. Now from this lesson, I'm going to turn to Biointron's single B antibody discovery. In Biointron, we call it AbDrop. This AbDrop is a single B cell screening antibody discovery platform. It's a microfluidic technology-driven droplet-based one. So the first step will be to get target B cells by sorting to get the positive B cells. To do so, we also begin with animal immunization, and then we harvest the B cells. So then we, by means of so-called cell encapsulation, we generate microfluidic droplet. Inside each droplet, there is one single B cell there, along with its target antigen and a secondary antibody and some other necessary bioassay keys. So then we will scan and look at each single droplet. And you may see that in each of the droplet, so once the B cells, after maybe cultured for a few hours, they will release the target antibody, which then will be captured by its target antigen and also by its secondary antibody. So this trimer complex will trigger a so-called threat. So the threat will release a red fluorescence, as shown in the lower right. So this red fluorescence will then be detected, and this droplet will then be sorted because there is a positive drop. But in case there's no cells there inside the drop, or there is a B cells, but it failed to produce any antibody, or it did produce antibody, but it's not a target antibody, so there will be either no fluorescence or just green fluorescence. So in both cases, they will be treated as a waste. Then we will sort all of those positive droplets as a pool. So this may sound complicated, so let's take a close look by zooming into the drop. So as I said, inside the drop, there is a single B cell there, which after a few hours culture may release some target antibody. This target antibody will be captured by the green dot, which is a target antigen, and also by secondary antibody, which will bind to the FC part. This complex will trigger a threat, and then this droplet will release a red fluorescence. This is The left one is for free antigen, some pre-antigen. But in some cases, like GPCR, you don't have proteins. Then in this case, we will use cells. So along the cell membrane, we have targets there. So this target antigen will also bind to the target antibody, and also second antibody will bind to this complex. So this trimer complex will similarly will trigger the FRET, and then the red fluorescence will be released. And then we will sort all of those positive droplets as a pool. Then we will reach the second step. In the second step, in each microwell, we put just one single cell there, which is paired by a barcode. In the barcode, a bead. In the bead, there's a unique barcode is there. Which the unique means this barcode sequence is unique. And then we will last the cells to release is mRNA, which will be captured by the barcode. Then we, in the magnetic field, we will retrieve the beads along with the mRNA, and then we do RT to get the cDNA, and then we do NGS to get this antibody encoding sequences. And then after a few bioinformatics processing, we got paired heavy chain and light chain. So you may wonder, how can we know they are paired? They're natively paired heavy chain light chains. We know this by looking at their barcode sequence. If they share the same barcode sequences, then we'll say they are from one same B cell. So this slide give you some numbers just for you to better understand this platform. So after B cell sorting, say in this case we got more than 8,000 cell hits, the so-called positive droplet, and then we release mRNA and cDNA, got cDNA, and after NGS and after bioinformatic analysis, we got around more than 2,700 paired sequences. Among these paired sequences, after we remove some repeated ones, we got more than 1,600 sequences that are unique in its amino acid sequence, in the variable amino acid sequences. And among these, there are around like 400 sequences that are unique in their CDR sequences. So from this around 400 sequences, we may pick up some top ones for our further screening. And then, yes, as I said, we may pick up some top ones. Then we got their purified samples, and then do some binding and functional screening, and then we got the best ones. So from what's introduced and what's going to share in the coming slides in our case study, you will see that our Biointron AbDrop holds some key features that do help us to go faster and better in antibody discovery. First is this fast track approach. So I may not have yet mentioned. So we can screen up to 2 million plasma B cells in just one day. And once we got those positive B cell hits, from B cell hits to paired sequences, just 1 week. And from sequences to purified antibody samples in just 2 weeks. So that means in general, from shots to verified hits, just 2 to 3 months. So it's a very, very fast. And second is since we are mining the whole plasma cell population, we are maximize antibody CDR diversities. This is very important for some challenging targets and for some advanced functional binding antibody requirements. And this AbDrop platform is also good not just for mouse. It's also good for rat, rabbit, and some fully human transgenic mouses to meet different project goals, which will be shown in the coming case studies. Yeah, so I also pick up some representative ones and some challenging targets to show how this single B AbDrop antibody discovery approach help our customer in their antibody discovery-based project. First, let's also begin with a very challenging one. It's also is a GPCR 7 times transmembrane one. So as I said, is they have low immunogenicity, very limited antigen choices. So in this case, we use DNA plasmid plus cells to immunize the mouse. And then we got their B cells, and then after screening, we got 177 paired sequences, and from them we pick up 78, and then we found 30 Phage binders, and among them 6 of them are blockers at the cell level. So you may wonder why the paired sequence number is not so great. It's because this target is GPCR. They are of low immunogenicity. Secondly, also GPCR. This time we use better choice. We have better choice, which is mRNA, and we also got around 250 paired sequences, and we pick up 70 to express, and then we got 25 Phage binders, and among them, there are 20 that cross-bind to cyanotaxis, and amazingly, there are 13 that also cross-bind to mouse in cell-based level. And the third case is peptides. So as you may know, peptides, they are of a kind of small size, like just a tenth of amino acid long. So their immunogenicity is low due to their limited epitope and simple structures. So we conjugate this peptide to some carriers like KRA, KRH, or BSA for animal immunization. And after B cell sorting, screening, we got more than 400 paired sequences, and from them we pick up 51 and to take a closer look. Among them, we find 37 ELISA binders, and in cell-based level, we got 17 blockers. Case number 4 is not antibody discovery from mouse. This time it's from a rabbit. So because in some cases people may want to get some antibodies of better affinity. So mouse is also, although it's most often used, but the affinity is not always good. So when you need some better affinities, you can tend to rabbit. Or if your target is mouse, then you may tend to some other host other than mouse. So in these cases, the target is a virus protein. So we, from a rabbit, we raised plenty of antibody hits, and most of his shows a very good binding activities. I would say most of them are like 100 better than the benchmark. This benchmark is mouse antibody from hepatoma. And case 5 is a small compound, and also this antibody is raised from a rabbit. This compound actually is a payload of ADC. So as you know, ADC, they are non-protein, non-peptide. They are just some chemical compound. So their immunogenicity, I would say, is super low. So then we had to conjugate it to some carriers like KH or BSA. And after animal immunization, B cell sorting, screening sequences, we totally got more than 1,300 paired sequences. And among them, we pick up 40s, and 29 are showing very good binding activities. And this one is not a mouse, not a rabbit anymore, it's a rat. So just to show you that our AbDrop platform is also good for raising antibody from a rat. And that's not everything. We can also raise antibody from transgenic mouse, like fully human transgenic mouse. So as many of you may know, transgenic mouse, after those genetic, since they are transgenic one, their immune response may not be super strong. And if the target is of low immunogenicity, so if you try to raise antibody with the platform like hepatoma, you may fail. So as shows in this case, this target is human protein X. It shows very high sequences with mouse counterparts. I would say it's more than 90%. And our customer did try to raise antibody with hybridoma, with hybridoma, but actually he only get just 2 binders. So when it came to our platform AbDrop, we were able to to raise tons of binders. So and most of them show very good binding activities. Yeah, but I finished this last case study. Actually, I finished my introduction of Biointron's discovery platform. So from the slides, you can see that Biointron's Phage Display VHH platform is a proven system for single domain antibody generation. And our AbDrop single B cells screening platform is a very powerful one for IgG antibody discovery. Actually, in addition to antibody discovery, Biointron also offer service like leads engineering, antibody humanization, optimization, and some drug-like track assessment. So we are trying to offer our service to fully cover those early stage steps. So as you can see from my introduction, these cases, Biointron has always been dedicated to science-driven innovation. We have been closely follow the antibody industry bottlenecks like the need for high speed, need for high throughput, the need for better antibody CDR diversities, the need for better, more specific bindings, or some desired antibody binding biases, and some advanced functional requirements, or some unique requirements. So we are continuously advanced our platform to better address these real-world discovery challenges. So every project either shared here or not reflects Biointron's mission to transform antibody science into design antibody hits. By doing so, we empower our partners all over the world to move from idea to therapeutics faster and smarter than ever before. Thank you, everybody. Thank you for your time. I'm be glad to take any questions.
All right, wonderful. Thank you very much, Dr. Liu, for that great presentation. Perhaps we can start with a question that came in when you were discussing the case studies, and that is, how do you decide the panning strategy?
Oh, this is a very good question. So to choose panning strategy, we will, as I said, we align the targets, our campaign goal to design this. So say if your campaign goal is to get some protein binders, we may just do some solid panning, because the protein binder should be good enough for your project. But if your project is for drug, so we may have to get some cell binders. In this case, we will first rely on cell panning, or at least liquid panning. So in this case, the solid panning will not be the best choice.
Okay, fantastic. You talked obviously a lot about the different animal hosts. For the single B antibody discovery platform, what sorts of key factors do you consider when you do select the hosts for antibody generation?
Oh, that's a good question. Actually, our customer choose which animal host. So say in most cases, mouse will be the first choice, because it's established. So it's technology. But if your target is from mouse, you may tend to rabbit or rat. Or if say I'm going to have a PK study tools. So in this case, for this to meet this need, you may better to tend to rabbit, because antibody raised from rabbit, they are of much higher affinity than mouse. Or maybe you may tend to rat if your target is from mouse, or if you want to have a better yield. So because antibody from rat, in many cases they have better yield. So in this case, you may tend to eat. Or if you have a concern about the ADA, you can tend to transgenic mouse. So, and also although I just show that our single B platform is good for mouse, rat, rabbit, and transgenic mouse, actually we are also working on some other rare species like alpaca, and horse, and chicken, and maybe in the future, yeah, you will see them.
Okay, fantastic. So it depends a bit on the project, the goals, etc., and so on. Excellent. You talked obviously about the phage discovery and the single B cell. Can the two platforms be combined in some way? So, for example, using a single B cell derived hit for an input for phage discovery.
Oh, this is a fantastic question. Yes, actually, these two kind of platform have been combined, although not yet in Biointron. So, say you got some positive B cell hits from your single B screening, and then after NGS, you got their paired sequences, but your final drug format may not be IgG. You want to turn to SLV or Fab, or if your antibody is read from transgenic Havigen only mouse, then you will have VHH. So in all of those three cases, you may display this sequence in phage and then continue with phage system to finish this discovery. Yes, this can be combined, although it may have not yet been factored in Biointron.
Okay.
We can do so. Yeah, if you do have those kind of needs, we can do so. Yeah.
Okay, great. In this question here, they ask for targets where purified protein antigens are not available. What other antigen formats or strategies can you employ to get successful antibody discovery?
Yeah, that's a good question. Actually, in the first few years, we did struggle in this part. For some hard to make targets like GPCR, like CD20, Claudin 18.2, since there's no protein material, you have to tend to DNA, plasmid, viruses, cells. But each of these, they have their problem, like efficiency, transfection efficiency, their efficiency for in vivo translation. Finally, we tend to actually, I would say, in the recent years, mRNA is our first choice. mRNA only, or mRNA plus VLP, or mRNA plus cells. After round 2 shots, we will check the titers. So if mRNA works well, we will stick to mRNA. Or if you do care about the cell binding, we may choose to add the final rounds to both animals with either VLP or cells.
Okay, excellent. Do you have any non-immune library antibody discovery services?
Oh, yes, we do. And also, I see that there is a question also raised here to say, can naive library also be used for screening?
Mhm.
Actually, we do have some for VHH. We have a semi synthetic, naive, fully human library, which now is just ready for service. So we combine hundreds of naive animals. They are CDR3, and also we collect some ready pre-existing CD1, CD2 sequences, and after some modification, we have already constructed this synthetic naive library. Now this library now is ready for service. Yeah.
Okay.
Also,
excellent.
yeah. So I just want to add something. If you happen to have some naive sample, like some from human, we can try to use our single B. But as you know, those target B cells, they are not enriched. The titer B cell may be very low. So I would worry about the sorting efficiency, even though we can try, but the sorting efficiency should be a concern.
Understood. Okay, thank you very much. We've talked the audience polls, the audience interest was about timing as well. what is the typical timeline from an antigen to a lead candidate?
Oh, yes, that's good. Good question. For our VHH Phage Display platform, generally takes from material preparation to validated hits, generally 4 to 5 or 6 months. Yeah. But for our single beach, it's much faster. As I said, from shots to hits, we can finish as fast as 2 to 3 months because the immunization take around 1.5, and sorting NGS just 1 or 2 weeks, and antibody production and validation maybe 3 or 4 weeks. So in total, just 2 to 3 months for our AbDrop single bead platform.
Okay, excellent. And how do you ensure, the second part of this question was, how do you ensure efficiency and high success rates as well then during that time?
Oh, that's good question. For phage display, as I said, we will align the antigen with our the target with our antigen design, immune strategy, and panning and screening approach. So we will based on the targets. So the target means antigen and target antibody, which is what kind of antibody you are gonna obtain. Then we'll design. Say in the immunization part, we will try to do something to protect the possibility to get high enough titers. And once we get the PBFC samples, we will try to do RT PCR in a high efficient way. Try to not to lose many B cells, which is to help to protect the B cell clones diversity. And in the panning part, we may not try to begin in the round one in the too stringent solution. So kind of mild followed by some stringent panning screenings, washing screening solutions. For single B platform, actually, I would say similar story. We will follow the target specific solution, and also with this AbDrop technology, this will help us to protect the B cells clones diversity, and each step we will try to control the false positive rate. Finally, we will get I would say the B cell clones are CDRs of the maximal diversity.
Okay, excellent.
Also our fast track antibody production just from sequences to purify antibody just 2 weeks. It's also help our customer help us to accelerate this project.
Okay, excellent. We could probably squeeze in a couple more questions here. I'm not sure if you see any that you'd like to that have come in. There's one here about the flexibility of your models. So fee for service, how flexible your collaboration models, fee for service, milestone, co-development, etc.
Oh, currently in most cases is a fee for service, and for some challenging targets, if these targets is kind of the first time we tried, we may set some milestone for our customer to finish the payment, but overall it's still a fee for service. So we have not yet tried some co-development pattern. The reason why we do so, because as I mentioned in the very early beginning, Biointron is a CRO. We try to do everything that we can to make our customer IP issue concern free.
Okay, excellent. I think perhaps then, if in the interest of getting everyone to their next appointments on time, I think we'll end there. But I'd like to say a huge thank you, Dr. Liu, for joining us today. It's been a real pleasure having you with us. A big thank you also to everyone in the audience for participating. And in closing, we hope you enjoyed the Scientist.com and Biointron webinar, and we hope to see you again next time. Thank you, everyone.